Well, good morning, everyone. Welcome to our second in our webinar series, From Bench to Bedside, the Promise and Hope of MSA Research. Before we start with the program, we have this to read to you. This educational session is not intended for the purpose of providing medical advice. All information, content, and material is for informational purposes only and are not intended to serve as a substitute for the consultation, diagnosis, and or medical treatment of a qualified physician or healthcare provider. The information supplied through webinars on our webpage or by any representative or agent of the MSA Coalition, whether by telephone, email, letter, or other form of communication, is for informational purposes only and does not constitute medical, legal, or other professional advice. So we want to let you have that as our opening, and we welcome you all. As I said, you're joining our second in a webinar series, and we'll get started here. This series is designed to highlight researchers who have applied for our seed grants. We have been providing seed grants since 2013, and through our last year of awarding the grants, we have awarded 49 MSA Coalition seed grants to researchers for a total of about $2.5 million. We want the grants to look at 4 different areas. They would be pathogenesis, biomarkers, preclinical and clinical treatments for the disease. And these are all submitted each year. The proposals go through a rigorous review process. The review process is done by researchers who have volunteered to help us with their time and their effort. The finalists are approved by the Coalition Scientific Advisory Board and then ultimately by the MSA Coalition Board of Directors. Everyone in this process is dedicated to finding a treatment or cure for this disease. We all have skin in the game. So today, with this second one, we want to start out by having everyone get introduced. And the first person to be introduced is our guest for the day. He's a researcher that was at University of Pennsylvania when he was awarded the grant. And we've asked him to join us, and he has decided it would be wonderful to let you know more about his work. And so first of all, we'd like to introduce Dr. Peng. So Dr. Peng, if you'd like to produce or share a short bit about your life before we get started and go further, we'd appreciate it.
Okay, yeah. So good morning, everyone. So it's really a pleasure to join here. I was originally trained in China as a kind of basic scientist who studies neuroscience. Then I think I want my research to be kind of benefit the patients and the society more. So I kind of switched the field of neurodegenerative disease when I come to the US. So I was training with one of the leaders in the field, whose name is Virginia Lee, at University of Pennsylvania. It's there that I started to get interested in multiple system atrophy. And then I kind of have this very good experience with the MSA Coalition. They support our research with the seed fund, and which turns out to be a very important discovery for the field. And then later on, I become an independent scientist, build my own lab at the University of California in Los Angeles, and started my own research here and continue to focus on multiple system atrophy.
All right, thank you, Dr. Peng. And we'll learn a lot more about he and his work as we move through the day. We do have 2 of the 3 who have been working on this webinar series with us today. One was unable to make it because of work requirements, but we do have one of the other ones to join us, Ariana Pitaro. And Ariana, if you'd like to share a short introduction of yourself, we'd appreciate it.
Sure, yeah. So I'm really excited to be here today, and I've been working with the MSA Coalition for several months now. I work at Brigham and Women's Hospital with Dr. Vic Khurana doing MSA research. So it's something that's very important to me, and I'm just excited to hear this webinar today.
Thank you, Ariana. And my name's Larry Kellerman. I'm a member of the MSA Coalition Board of Directors. Been working with the board since 2014. And we decided about a year ago to begin putting together a webinar series that would highlight these seed grants that have been awarded to the various researchers. And I was only so happy to be able to be a part of this. So I'm glad to be here today, and hopefully we'll be able to get a lot of information and learn more about not only Dr. Peng's research, but also one of the, the protein that seems to be such an issue for MSA, and that is alpha-synuclein. So the way the grant works, Dr. Peng's topic was in the area of pathogenesis, and pathogenesis, as it's defined, is the process by which a disease or disorder develops. So the pathogenesis would look at the beginnings of the disease as well as the course or journey of the disease toward its end. And so that's where his grant was applied to, and through the process, he won the $50,000 to do his research. And so right now, Dr. Peng, if you would like to begin sharing, we will hopefully be able to learn a lot from the work that you've done over the past 4 or 5 years.
Sure, yeah, absolutely. So let me start share my screen. Could be this one share. And let me start to. Okay, could everyone see my screen now?
Yes, sir.
Yeah, first of all, I really appreciate this chance to share my research here. I have given many scientific talks, but I think this is a very unique and maybe the most meaningful one, because in disease, I was always hoping that our research would have a kind of impact on the patient and caregivers to really benefit the society. So I'm really happy to talk with this audience. So my talk today is really about like alpha-synuclein, which is the pathological protein for multiple system atrophy. So let's start with a little bit of background. As many of you may know or may not know that like many of the neurodegenerative disease, which includes like Alzheimer's disease or Parkinson's disease that most people may know, are kind of characterized by the accumulation of disease proteins in the brain. So you may ask me, what is really a protein? So you may know that your body is made of cells, and the protein is really a key component and maybe the most important component to make up a cell. So it is a kind of a very basic component for all the life on this earth. So these proteins, they have their normal functions in your body, and they make you be able to perform the task that you want to do. And however, under disease conditions, some of these proteins could go wrong. They go wrong in a way that they change their shape. So they are not in their normal shape and cannot perform their normal function. They become this disease shape that make them to start to bind with each other. And once more and more protein bind with each other, like millions or trillions of them bind together, you can see they will form the very, very big protein complex in your brain. So this, you can see this brown color here. This is kind of detect of a specific protein in the patient using a reagent, what we call antibody. So antibody is really kind of the reagent that people usually use to detect any specific protein in your body. So in this particular case, in Alzheimer's disease, this is using an antibody to detect a protein called A-beta in the brain tissue. And this purple and this blue color is the nucleus of your cell. And this brown color is really the very big protein complex that formed by this A-beta protein. And also in Alzheimer's disease, you can see this is a nucleus of another neuron. And this neuron is filled with this protein called tau, which is in the brown color. So these are the kind of very characteristic pathological feature of the disease. And for Parkinson's disease, it is another protein. The name is called alpha-synuclein. And if you use antibody against alpha-synuclein to detect it, you could see that it forms this very big kind of protein complex in this neuron shaped here. And you can also see that actually many of the neurons in the brain don't have it. Only a group of neurons have it. So you may ask them, why should we care about this protein complex? So the reason is that there's a lot of evidence support that these pathological proteins, or these disease proteins, these protein accumulations is really the cause that play a key role in the development of this disease. So if we really want a therapy to slow down or reverse the disease progression, we should try to deal with this disease condition, which is this protein accumulation. So during the past decades, there's people find one very important feature of this disease protein, which is that many of these disease proteins, you can see that they are in this very unique shape that is different from their normal shape. So what they can do is that this disease protein could use itself as a template and recruit a normal protein and then change the normal protein to this disease shape. So using alpha-synuclein as an example, the disease shape alpha-synuclein could use itself as a template to recruit normal synuclein and change the normal synuclein to the disease shape. So you can imagine if they repeat this process many times, the disease-shaped alpha-synuclein will build up in the cell. There will be more and more of it. It's like a virus that amplifies in your brain. And eventually they form this very big protein complex that will disrupt neural function and eventually cause the disease. And more importantly, this disease-shaped protein could not amplify the cell, but they could also kind of jump from one cell to another cell. So this is one cell, this is another cell. The disease protein could kind of transmit from one cell to another one and amplify the cells in this recipient cell again. So this is very like kind of a virus infection process, right? You get more and more cells in your brain that is affected by this disease protein, which continue this amplification process. And eventually the brain is filled with this disease protein. That's why the disease is getting worse and worse every year. So, so back to multiple system atrophy. So for multiple system atrophy, actually the pathological protein is also alpha-synuclein. So it actually shares the same disease protein as several other diseases, including Parkinson's disease, dementia with Lewy body, and Alzheimer's disease. However, multiple system atrophy is very unique. The reason is that in all these other diseases, the disease alpha-synuclein is accumulated in a group of cells in your brain called neurons. So basically, your neuron is composed of many different types of cells. One key component is called the neurons, which is the cell that is really perform the function that controls your hands, that forms your memory. But there's also kind of other groups of cells like this one, in the blue. This is called glial cell, sorry. This is called glial cells. So a glial cell is like a helper cell that help to support the neural function, but it is also critical for you to have a normal functional brain. So while in all the other diseases, this disease synuclein accumulate in neurons in here. In multiple system atrophy patients, vast majority of disease synuclein actually accumulate in this glial cell called oligodendrocytes. So that's why MSA is so unique, that is very distinct from the other diseases, even though they share the same disease protein. This is also the reason why we are kind of so interested in MSA, because we think this is a unique disease. like that is a kind of shared mechanism with our disease, but also have a very unique aspect by its own. So then we try to ask a question, like why? Like why is this the same disease protein will exist in a different population of cell in multiple system atrophy compared with others? How could this even happen? So we think, like we build a kind of hypothesis, or we think one possibility is that because when synuclein changes shape to this disease shape, maybe there's multiple different types of disease shape, that these different disease shapes could have different properties that eventually lead to their different distribution in neurons and glial cells. So to help you better understand this idea of different shapes, I kind of borrowed this idea from my student. I don't know how many of you have played this game called Plants and Zombies. And you can imagine that these different shapes of disease synuclein are different type of zombies. They're all going to damage your brain, but they will damage it in very different ways.
Dr. Peng, before you go further, is MSA cerebellar and MSA Parkinsonian both seen with that same general straining of alpha-synuclein?
Yeah, that's a kind of very good question. So I would say they all have these characters. So for both MSAP and MSAC, they all have synuclein aggregation in oligodendrocyte, in glial cells. They share that. But it seems like the origin or the first brain region that affected by synuclein is different in these two patient populations. However, we haven't really identified any detectable difference in terms of the shape of synuclein in these two types of patients.
Okay.
Yeah. So here is our kind of hypothesis. We think MSA and MSA disease alpha-synuclein have a unique shape. We call this GCI alpha-synuclein. And well, in all the other disease, when synuclein accumulated in neurons, we think it's a different shape, and we call it Lewy body synuclein. And we think the GCI synuclein shape will more easily to induce this protein aggregation in glial cells, while the Lewy body synuclein is more easy to induce this protein aggregation in neurons. So which is the reason they have different distribution in this different patient population. And then we start to test this kind of this idea to see whether it is really true. So we first kind of develop a protocol to purify this diseased protein from the patients. Like you can see from here, this is the disease protein we get from the MSA patients and from the PED patients, and you couldn't see this population of protein from a normal patient. So then we start to ask whether they indeed have different shapes. So this is a kind of biochemical experiment that I won't go through details, but you can kind of see the bands here, right? That this Lewy body synuclein, they have different bands compared with GCI synuclein in this test. So this different bands is really a strong evidence demonstrate that they indeed have different shapes. And then we want to see whether they have different properties to induce disease. So we kind of establish a method to culture these glial cells that is affecting MSA in our culture dish, in a dish. And then we treat them with the disease material, disease synuclein we get from the patient. And then we want to see how this patient material will affect these disease-related cells. So like, as you can see from here, this red color is really an indication of kind of, you can think about it as a toxicity to this disease, to these glial cells. And the more red color you see, means that the material is more toxic to these type of cells. And you can see that the material from MSA patient induce so much of this red color in this culture, in these cells, for both MSAP and MSAC. But for the other disease materials, there's a much less red color here, suggesting that they are much less toxic to this group of cells. And if you do a careful quantification, you could estimate that the material, the disease synuclein from MSA patient is roughly about 1,000-fold more toxic to this group of cells compared with the disease synuclein from PD, AD, or DLB. So which we think is kind of very important, contribute significantly to the pathological feature of MSA.
At this point, Dr. Peng, did you have an idea of what was causing the toxicity, or how the toxicity was impacting the cells? Is that something that was already part of your understanding, and then you were taking it farther along in the process?
Yeah. So to the first point, what is the how it induces toxicity? So yeah. So I said toxicity here to kind of simplify the effect, but it's actually kind of induction of synuclein aggregation in these cells. So it's basically like sorry, I have to go all the way back to show. Yeah. So it's basically doing this process in that cell. So you put this diseased protein from the patient into the cell, and once you get into the cell, you start to amplify the cells. So the red signal is kind of far away. The red signal we see here is the amplified diseased protein in the cell, which means it starts to affect the cell. So it's to the second question, like, do we have any clues about what is the kind of toxic effect it have on these cells? That's actually a really kind of important and interesting question. We are kind of doing more experiments right now in my lab to answer that question. But to this point, we haven't really done anything to explore the detailed toxic effect on these cells by this pathological protein.
Okay, thank you.
Mm-hmm. So that this is kind of consistent with our kind of guess, which is that GCI synuclein from MSA really affect these glial cells dramatically. Then we want to see that Lewy body synuclein really affect neurons more than the GCI synuclein from MSA patients. Basically try to show this part. So we grow these neuron cells in the dish and treat them with the material from MSA patient or from PD, AD, or DLB patients. And as you can see from here, again, the red color indicate the amplified disease synuclein in this group of cells. However, you can see that there's so much more red color in the MSA patient material treated cells compared with PD, AD, and DLB treated cells. That means the disease synuclein from MSA patient is also so much more potent to induce neuron disease in addition to glial cell disease. So this is in kind of in contrast to what we think it could be. So we think this idea is wrong, actually. So because this observation is so surprising, we actually verified many times with material from so many different patients, and you can see the result is so consistent that the material from MSA patients is always so much more toxic than the material from other diseases. No matter you test it in glial cells or in neurons or in a kind of cell that's unrelated to the brain, kind of unrelated cell line. Right. So that it seems like the material from MSA patient is a generally much more toxic material that don't show any cell type preference. That means you don't preferentially affect neuron or glial, but it's just much more toxic than the material from other diseases. So we think this hypothesis is actually wrong, that it's not like their property kind of make them preferentially only affect one group of cell, but that this strain is just more toxic. So then it raises a question: if this is the case, then why this very toxic synuclein strain always exists in oligodendrocytes in multiple system atrophy patients? Like why they have this correlation between the cell type with the toxicity, if the toxicity itself don't have cell type preference? So then we kind of have a different idea. We think because these glial cells and the neurons have different intracellular environment, okay? We think maybe there are different environments make synuclein, disease synuclein to form different shapes. So that the glial cell environment always make the disease synuclein to form this very, very toxic shape. That is, we think that could contribute to the very aggressive nature of MSA disease. So then we think, how could we demonstrate this is the case? So we build a new mouse model. This is kind of complicated, but to make a long story short, that in this mouse, the only type of cell that could develop synuclein, disease synuclein, is the glial cells. So we kind of restricted that to the glial cell population in these mice. And then we kind of inject some of the disease synuclein that we generate artificially in the lab. It's not from patients, it's by the artificial disease synuclein we generated. We inject that into this new mouse model. And as we expected, they start to induce this disease synuclein accumulation in this group of glial cells, as shown here, the brown color. So that we have a glial cell, we have a group of disease synuclein that's accumulated in glial cells, but they come from artificial synuclein, disease synuclein. It's not from the patient. Then we prepared this disease synuclein from glial cells. We take them out, and we test their toxicity, and compare that with the original material put in there. And very importantly, you can see that once these disease synuclein start to accumulate in glial cells, they become so much more toxic than the original material you put in there. This is a very strong evidence demonstrate that something in this glial cell environment really make the disease synuclein much more toxic than they used to be. And then we further kind of verify this with other seeds. I won't go detail to that, but then we try to see whether we have find more evidence to support this, to make sure that we get it right. So we cultured and we growed all these different type of cells in the dish. Like this is oligodendrocytes, glials. This is the growth of neurons. This is the growth of another type of neurons. This growth of a kind of unrelated cell line. And we put the disease synuclein into them. So this is again the disease artificially generated disease synuclein. It's not from patients. And then once they induce pathology, like a synuclein aggregation in these different type of cells, we prepare it out, take it out, and we compare their toxicity. And you can see that the synuclein that is grown in this glial cell population is so much more toxic with so much more red color here compared with the synuclein that grow in other type of cells. And we also do a quantification here. So this again shows that this effect is very unique to glial cells, that only glial cells is able to make synuclein so much more toxic compared with the original seeds you put in there.
Dr. Peng, are you at this point, in the research, are you beginning to get an idea of what might be in the glial cells that is making these so much more toxic?
Yeah. You always ask these great questions. I will try to kind of address that in the next slides, actually.
Okay.
Yeah. So then I kind of just ask what Larry has asked it, like why? Like why would this happen? What exactly in oligodendrocytes, in glial cells could make synuclein more toxic? So this is actually a kind of important question, but it's super difficult to ask because you are comparing glial cells versus neurons, which is two fundamentally different cell types that could have thousands or millions of difference. How could you ever know which one is contribute to this property? Then we decide that we want to ask us one simple question first, which is that this glial cell, they kind of have very unique structure that the rows around the neuron like to protect the neuron. Okay, so we ask one simple question: does this unique structure of glial cell contribute to the development of this unique disease-synuclein shape that is so toxic? So we culture these glial cells and we generate a cell lysate. So we basically break them up to release the content from these cells. And so this is what we call the cell lysate. And you can see that in these break cells, all these structures are disrupted, but all the content or the factors in these cells are preserved. And then we put a synuclein protein in them to generate a disease synuclein in this different cell content from either glial cells or neurons. And you can test their toxicity. And you can see that the synuclein, disease synuclein that generate in this glial cell content is much more toxic compared with the synuclein that generate in this neuron content, which means that there is a factor in glial cell is independent of its shape or cell organelles that really contribute to the development of this disease, this unique shape of disease synuclein in MSA. So we think this factor is a key for kind of why MSA is so aggressive in nature compared with the other diseases. So if we could find this factor, we could target it and try to prevent the development of such a toxic disease synuclein conformation in MSA patient. So we think it's a very promising drug target. But right now we still don't know what this factor is. So my lab is actively performing experiment to screen for this factor. And it's a kind of a research that's ongoing right now in the lab. And then we kind of finally kind of want to check the opposite aspect, which means if this glial cell environment makes synuclein become so toxic, what if we move this group of these synuclein to neurons? Would the neuron change it back to the kind of less toxic synuclein conformation shape? So that could also be benefit. So what we did is that we amplified this material from the MSA patients in the neurons, in the neurons that we grow in the dish. And we check their potency after this amplification to see if we amplify them in neurons, could we reduce their toxicity? However, unfortunately, it seems like this very toxic conformation, once it formed, it couldn't be changed back to next toxic component, even if you amplify the in neuron culture.
Man.
So that's the kind of the last conclusion we get from this study. So, so this is more evidence to support that after amplifying this material from MSA patient neurons, they kind of maintain their property. You can see their bands is the same and different from another disease. So that means the neuron don't have the power to change them back to the less toxic shape. So with that here, I want to summary come out the key information we want to take home from here. The first is that we demonstrate very clearly that the disease synuclein MSA is very unique. It's so much more toxic than the disease synuclein we see in all the other diseases like Parkinson's disease, dementia with Lewy body, or Alzheimer's disease. So it's very unique, it's super toxic, which is very likely contribute to the aggressive nature of MSA. And then the reason why they're so toxic is because the unique environment in the glial cell where they formed that make them to become this very toxic shape. And then we know it's kind of a factor in these glial cells that make this happen. And we are in the process to searching for this, what exactly this factor could be. And then finally, if you move this material to neurons, neurons still couldn't make it less toxic to the patient. So that's the take-home message from here. And finally, I want to acknowledge like the people that really contribute to this work. These are the students that have been working with me. And I want to particularly thank my mentors, Virginia Lee, which is here, and John Chojaski, who is here. So these two is really my postdoc mentors and who really give me a lot of help during this process. This is actually a picture we take at UPenn when we do a 5K running to raise funding to support the research on neurodegenerative disease. And many of our collaborators here, and also very importantly, the support from the MSA coalition to support us for this research. Cool. I think that's it. Thank you so much.
Well, before you close it down, could you go back to the last slide? I had a couple of questions.
Sure. Yeah.
So your third point, they don't show any cell type preference. And so you say, therefore, the distinct cell type distributions is not due to their strain properties. Do you have any other hypotheses, or are you looking at other hypotheses to see what property or properties might be the issue here?
Yeah, we think like the expression of normal synuclein in the glial cell in MSA patient is, we think that's really a key. So in a normal condition, like in normal brains, synuclein, the normal synuclein barely exists in glial cells, which we think is the reason why in most patients like PD, like Parkinson's disease, dementia with Lewy body, this disease synuclein only accumulates in neurons, because for the disease synuclein, you amplify the cell, it leaves the contribution of normal synuclein as a substrate for them to amplify themselves. But in most of the cases, we think the glial cell don't have synuclein. So that's why in most patients, they don't go to the glial cells. However, I think for whatever reason, that in MSA patients, this synuclein protein exists in glial cells. It could one way or another. One possibility is that under this disease condition, this glial cell start to express or make this normal synuclein by itself. Another possibility is that in MSA patients, these neurons start to give their synuclein protein to the glial cells, which is a transport process that transport from neurons to glial cells. And that's another possibility. But the eventual consequence is that there is normal synuclein start to exist in these glial cells that support the amplification of disease synuclein in this glial cell population. We think that's the reason why they start to accumulate in these glial cells in MSA patients.
So it is partly the environment, partly what is being introduced into the environment of the oligonucleotide. Okay.
Yeah.
Now, Ariana, there was a couple of questions that we had come up with before that maybe would be good if you would like to ask one or two of those questions, just to see if we can get a better clarification on a few of these things. You want to ask? Okay.
Yeah. So, Dr. Peng, one of the questions that we had was, so why is the alpha-synuclein that we find in glial cytoplasmic inclusions, why is the structure more compact than the alpha-synuclein that we find in Lewy bodies, for example?
Mm-hmm. Yeah, that's a kind of a really good question, like why they have this different shape and why there is why they are more compact. The short answer, we don't know. We are looking for, as I mentioned, we are looking for the factors that in glial cells that could contribute to the formation of this very unique shape of disease synuclein MSA patients. But we still kind of didn't find that factor yet. We are screening for it. So we may have an answer for that maybe in the next couple of years.
Okay. I see. Okay. Thank you.
Mm-hmm.
Are you? Go ahead, Ariana.
Ahead. I was just going to ask a follow-up question. So, also, the alpha-synuclein found in GCIs with MSA is found to be more potent than that found in Lewy bodies. Do you know why there's such a, just big discrepancy in potency there?
Yeah, I think it's kind of related to the shape, because they have different shapes or different conformation. We think the different shape they have give them different properties. We actually have an experiment to show that we can generate a very purified these disease synuclein from MSA and Lewy body, show that they still have this very different toxic effect, which means it's not kind of the other proteins that with that brain acid that contribute to it. It's really the property of the synuclein in the cells that lead to these different properties. So we think the only explanation is that they are different structure or they are different shape that make them to have these different properties, which is the reason that it is more toxic in MSA.
Thank you.
When we've looked at these pictures, Dr. Peng, and they're pictures people who are in the MSA community have seen many times before. In the way that they are seen, especially on your slides, they all seem to have a similar shape. Would it be that as you can get down deeper and deeper into the shape of it, we'd be able to see, for example, ridges, see distorted segments of particular alpha-synuclein? Like here, when we look at this, it seems like they're all somewhat similar. Because of the resolution that we're seeing it in as much as anything?
Yeah, that's a good question, actually. Yeah, I think that kind of confused many people. Like, this is all like a round dog-like shape. Why do you say they have different shapes? So actually, what you see here is not a single protein. What you see here is an accumulation of millions or trillions of protein that all bind together. Gotcha. So this is so this shape is only kind of reflect the shape of the cell. When we talk about shape, we are talking about the shape of the molecule, like each individual molecule when they kind of compose these big protein aggregates. So that couldn't be seen easily by this staining. You have to use some method to detect that, to show they have different shapes.
Okay. So another question that I know all three of us that were preparing for this were going to ask you, I'm sure, moving on to next steps, you shared a little bit of what you're trying to do right now in your lab. Do you see any further development in this in such a way that maybe, as you said, a drug company or a pharmaceutical company might be able to look at this and say, hey, we could look for a compound that might address the issues that you've raised with your research? Yeah.
So I think one of the key questions, wait, let me go to that slide here and start sharing. So one of the key questions that we have been working on, actually two of them. One is that what is this factor that lead to this development of this very toxic synuclein structure? So we are in the process of searching for that. Actually, it could be anything, right? It could be RNA, like ions or lipids or protein. It could be anything in the cell. So we have developed a series of experiments, and we narrow it down right now to most likely be a protein that interact with synuclein, specifically in glial cells, but not in neurons. So we have narrowed down to that extent. So we are screened for that protein. I think once we get that protein, that will be a kind of a very important drug target that the pharmaceutical companies will be interested to further screen for compounds that could target that interaction between synuclein with that protein. That could most likely prevent the development of this very toxic synuclein conformation in MSA patient. So that's one aspect. Another aspect we have been focusing on is the downstream biological consequence of this glial cell pathology on neurons, because to affect the central nervous function, you eventually have to affect function of neuron. But right now, there's very, very few lab that is focusing on how glial cell pathology, glial cell protein aggregation would affect the function of neurons. So we are trying to explore that. So if we can find a key mechanism how to affect neurons, maybe we could develop a therapy to prevent that toxic effect on neurons and reserve the function of the brain for MSA patients. So that's kind of a true focus of the lab right now.
Gotcha. And your further research, you had talked earlier a bit before or about your mentors, Dr. Trajanowski and Dr. Lee. We had a couple of years ago recognized Dr. Lee for her excellent work in identifying the role of the proteins that are impacting ALS, impact Alzheimer's, impact MSA, Parkinson's, et cetera. How did her background and her research inform you as you move forward in yours?
Oh yeah. Actually, Virginia is really the kind of the people that introduced me to this neurodegenerative disease field. Actually, she's the one who suggested me to focus on multiple system atrophy because she thinks this disease is so unique. Because she works on everything. She works on Alzheimer's, Parkinson's disease. So she thinks, oh, MSA is such kind of a unique disease that it is really characterized by this protein aggregation in glial cells. It's not in neurons. So he said, child, kind of, why don't you think about this? And this could be a very important scientific field that there's not too many people that will kind of study this. And he think this is could be a very good topic for my scientific career. So I think that's kind of a very big influence on me and for me to focus on this multiple system atrophy.
And it's good that you have, because it is a rare disease. And I know that those people who are listening are somewhat, or who are like me, that have cared for an MSA patient or are an MSA patient right now. They know that there is a real need for a treatment to slow down, if nothing else, to at least slow down the progression of the disease. What would be your timeline if you were looking forward and, let's say, using the research that you've done and the work that you're continuing? What would you see as a timeline possibly for some sort of treatment to come out addressing what you've learned?
Yeah, I think it may be in the next, I would say, 3 to 5 years, then we will know the targets and maybe take another anywhere between 1 to 3 years to do a large scale screen. I mean, if the funding allowed us to do that, to screen for kind of the compounds to target those drugs, then we have something that could go to clinical trials to start to develop for this. That's kind of the. But I mean, like science is always like unpredictable.
Yes.
Yeah. So that's that. But if you want me to make a prediction, I think that's kind of the timeline that I will see be reasonable to fulfill this task.
Okay, and we've heard that from other researchers with looking at other fields as well, as a timeline of 3 to 5 years. And so for patients and caregivers right now who are just being diagnosed or have just been recently diagnosed, that might be coming along. For others, it's hoped that these treatments and potential cures will be there for the future MSA patients as well. So Dr. Peng, we want to thank you very much for sharing what you shared with us today, for the hard work you've done. I noticed, and it's on our website, that you had an article along with other authors published in Nature, which is a pretty good acknowledgement of the hard work you guys did. From that Nature article, did you get any feedback from others asking questions related to potential research they might want to pursue, or did that Nature article get further attention for what you are trying to do?
Yeah. It's kind of actually many of the focus the research focus that we have in our lab right now is a follow-up study of that Nature paper. And I kind of consistently get this question, like what this factor is. That's kind of the number one question I get whenever I give a talk. So that's why it's also why our lab is actively searching for that. But it's also kind of a very kind of risky thing because you have to try so many things to know what it is. But I think that's kind of the question that most people will be like very, very interesting to know. Like, it's not only kind of provide a very great drug targets, but scientifically for the whole neurodegenerative disease field, that will be the first factor you will ever know that will change the shape of a diseased protein. So that's why the whole field is so interested in that.
I would agree after reading it, even though I'm not very good at understanding all the science in it, that Nature article really did point to some interesting issues that I know you're addressing and others will be in the future. And I think 3 to 5 years, we hope that that'll be the case. There'll be a treatment available for people with this. Yep. Well, we want to thank Dr. Peng for sharing what he has shared today and the hard work he did. As you could tell, he did try to make sure that it was for patients and caregivers. We talked to him about it before then. And Dr. Peng, I think you did a very good job of making it as clear as possible. Your videos or your slides were very informational as well. So we really thank you for taking the time to do this and share with our patients and caregivers. And hopefully you'll be able to maybe in the near term apply for another grant from the coalition to help you move this research even farther along.
Yeah, sure. Yeah, thank you. Yeah, it's really a pleasure for me to share in here. As I'm really happy for your invitation. Thanks a lot for that.
Well, you're welcome. And we also want to thank each and every one of you who have donated monies to the MSA Coalition, whose monies also help fund these seed grants. Like I said, 49 grants have been awarded thus far, $2.5 million given out. And this year, we've already had the pre-proposals sent in. Those pre-proposals have been reviewed by our reviewers, and some of them have been sent back to be resubmitted as full proposals. And hopefully by our national conference coming up at the end of September, 1st of October, we'll have the announcement for those winners. And the next group of researchers who will do like Dr. Peng, try to find a treatment, a cure, an understanding of this disease. So from bench to bedside, that is the promise and hope of MSA research. We want to thank all of you for joining us. Ariana, I want to thank you for joining us and being with us today. And Casey, thank you for running the event. And just a short aside, for those of you who did not see any of us, there was a technical glitch, which would have meant we'd have to shut off the webinar and start it back up again. And we felt it was far more important to have you see Dr. Peng's slides and hear him and hear our voices rather than restarting the webinar. So hopefully that's okay with you. The last thing, next one will be in September, and the announcement of who will be forthcoming, and it will also be on pathogenesis. So we hope you can join us then. So again, thanks a lot. Thank you, Dr. Peng, Ariana, and Casey. Everyone have a good Wednesday and look forward to seeing you in a couple of months. Thanks.
Yeah. Thank you.
You're welcome. Thanks. Bye-bye.
Bye-bye.